Hello, everybody, and welcome back. Today, we're going to be talking about the process of cell differentiation, which has the word different in it. So, this is the process multicellular organisms are going to use to create all the different kinds of cells that do different functions in their bodies. Unicellular organisms do not use differentiation because they are just one cell, so they don't have these specialized cells that do different things for them. We talked about how egg and sperm come together in the process of fertilization to create that diploid zygote with all the DNA that that organism is going to need. And that zygote, that first cell formed after fertilization, is going to start to divide and create this mass of cells that will eventually be the body of an organism. And as these cells are dividing, they're picking up signals from their environment, where they are in this mass of cells. And those signals are going to determine what that cell is going to be able to do, what genes it's going to need to read. Multicellular organisms are organisms that are made of more than one cell. Multi means many, so these organisms have many cells. And all those cells need to work together for the survival of the whole organism. Remember that first cell formed after fertilization is called the zygote, and it's going to divide by mitosis to make genetically identical copies of itself. And that we're going to start getting a big ball of cells in this embryo, and where those cells are in that embryo are going to determine what those cells' functions are going to be. So multicellular organisms are not made of a single type of cell. They have many different types doing specific things. For example, we have cells in our lungs that are specialized to take in oxygen and get rid of carbon dioxide. We have cells in our stomach that are specialized to secrete acid and break down materials coming into the digestive tract. So multicellular organisms, many different types of cells, all doing different functions, but all necessary for that organism to survive. All multicellular organisms are eukaryotic, so they contain their DNA in the nucleus of the cell. And all the cells that come from that zygote are going to have the same genes, but they're going to differ in what genes they use based on the signals they're getting from their environment. And this is going to create special functions in a cell, which we call cell differentiation or cell specialization. And the cell appearance is going to be directly related to its function. So the genes that are red are going to cause these appearances that help these cells do a certain thing. Red blood cells, for example, they're tiny, they're round, and they can squeeze through these capillaries and deliver oxygen and take in carbon dioxide. White blood cells have these pseudopods for wrapping around pathogens and destroying those for the body. Nerve cells need to transmit messages over long distances, so these are very long cells with lots of branches for sending and receiving messages. And then we have sperm cells with the flagella, and this is the only human cell that has a flagella to help this sperm swim to the egg. So all of these cells that have pretty much the same DNA, the sperm cell only has half of it, but these cells are specialized to do a certain thing, and they all have the same DNA. So they're different in the way that they read that DNA. Like we said, all multicellular organisms are eukaryotics. They have their DNA contained within a nucleus, and that DNA is going to be wound into structures called chromosomes. And we can see here a sister chromatid, which is a chromosome, and its exact copy. And we're unwinding one of these chromosomes. It's going to be wound up tightly around these histone proteins. They're kind of like spools for this thread to be wound around. And when this DNA is wound tightly around the histone proteins, it's not accessible by the cell's machines. It can't be read. So when the DNA needs to be read, it's going to be unwound, and we're going to have this double helix that's free and accessible by the cell machinery. So these chromosomes are going to be divided into segments called genes, and these genes are sequences of nucleotide bases that are going to code for a protein or a regulatory function of the cell. And these different proteins are going to make up our traits, our characteristics. So those traits are encoded for by this DNA in this eukaryotic cell. We're going to find that DNA in the nucleus. We've talked about zygotes already. This is the first cell formed after fertilization. Zyg actually means together. So this is when egg and sperm come together to give us a diploid cell with two sets of DNA. And multicellular organisms are going to start out as zygotes. And this zygote, this single cell, is going to start dividing to give us the cells of the whole organism. This zygote is a mix of DNA between two individuals, a mother and a father. And that DNA is going to serve as the blueprint for the proteins and the regulatory functions of the whole organism. So all cells receive the same DNA because zygotes do divide by mitosis. But the cells are going to differ in the way they read that DNA based on, again, where they are. So we can see here the process of fertilization. You've got a sperm, and the enzymes in the sperm are going to dissolve this gelatin coating around the egg. And then the nucleus of the sperm is going to fuse with the egg cell and be injected in. These two nuclei are going to merge. And then we have our zygote with two sets of DNA, one from mom and one from dad. Though every cell that forms from that zygote is going to have the same DNA, they're going to read it differently. Cells only read what they need. Much like if I hand you a biology textbook and say, hey, we're going to have a quiz on differentiation tomorrow, you're probably not going to try to read the whole textbook. You're just going to try to study up on that one particular section about differentiation. So you become specialized with that knowledge to help you on the quiz. Cells are the same way. They have all of the DNA, the entire genome, all of the genes in the organism, but they're only going to read the parts to make the proteins that they need to make. They're going to ignore everything else and turn those genes off. So a stem cell can become a muscle cell by reading the genes that it needs to make muscle protein, while a cell for the blood, for example, is going to read only the cells that it needs in order to make protein to carry oxygen. This process of cells becoming specialized occurs in embryos, so you're born with all the different types of cells you're going to need in order to survive. Each stem cell is going to start with the ability to differentiate into a wide variety of different cell types. But once this stem cell has chosen a path to specialization, it cannot go back and change its mind and become a stem cell again. It's going to continue down that path of specialization until it becomes the target cell, whatever it may be. So this process is irreversible. Once this stem cell is made, say, a nerve cell, this nerve cell can't change back into a stem cell, nor can it turn itself into a red or white blood cell or a sperm cell or any other kind of cell. It is stuck being a nerve cell. Now, when we have these specialized cells, oftentimes they can reproduce by mitosis to give exact copies of themselves. So, for example, if we have a stem cell that becomes skin, and your skin becomes damaged with, say, a cut, then the skin cells around that cut can replicate by mitosis in order to fill that gap and heal the damage. But the skin cell cannot go back and decide to be a stem cell again. It's stuck being skin, and that's all it can make are skin cells. So stem cells are unspecialized cells that can become pretty much any kind of cell, or at least many different types of cells. There are two types of stem cells: embryonic, which come from embryos or developing offspring, and adult stem cells, which we find in pockets in mature adult organisms. And these adult stem cells are used for growth and repair to the body. So embryonic stem cells are a little bit more flexible. They can become more different types of cells than these adult stem cells can. These have already kind of picked a certain path, and they can become certain types of cells along that path. But both of these are extremely promising in medical research. Embryonic stem cells are frowned upon. We don't use them a whole lot because it does require destroying an embryo. But adult stem cells, we've had a lot of luck with taking these cells out of an individual that might have a genetic disorder, manipulating those stem cells so that they no longer have the gene for that disorder, and then putting them back into the adult that they came from. And this has been used to cure certain types of blindness, and is even being used to treat diseases like sickle cell disease. We've talked about gene expression. This is the process of making a protein from DNA. So reading the DNA to make mRNA during transcription, and then using that mRNA in the ribosome to make a protein during translation. So the organism's traits are going to depend on the types and the amounts of protein that it makes. For example, your hair is a certain color based on the type of protein you make. It's wavy or curly based on a different kind of protein that you would make and put in your hair. So how this DNA information is used to make proteins is called gene expression. Gene regulation is the process of controlling when to make protein, the type of protein to make, and how much of it to make. So not all cells express all genes. You don't make all the proteins that you could possibly make in every single cell. For example, we talked about how your heart cells don't need to detect light, so they're not going to make those proteins. Gene regulation is controlled by signals from the environment that tell a gene to turn on or off. And when a gene is on, it's producing that coded protein. And when it's off, it's not producing that protein. And some cells are going to turn genes off permanently, like your heart cells turning off that gene to make the protein for detecting light. They're never going to need that. So gene regulation is affected by the environment and signals from other cells. A little joke for you. We have a mature cell here, and he says, when I was a little stem cell, I didn't know what I wanted to be either. And your little stem cell, he's so confused because he hasn't picked his path yet. Certain cells we need to be able to recognize and know what they do. So these are the cells that we're going to look at in animals, and these are the cells we're going to look at in plants that have specialized roles. And we're going to look at all of these one at a time. Fungi also have specialized cells, but we're not going to focus on them in this class. Just know that they are multicellular and they do need these specialized functions. Nerve cells are also called neurons, and neur means nerve. These are very long, thin, branchy cells that are specially suited to their function, which is to send and receive messages to and from different parts of the body. The membranes of nerve cells have these sodium-potassium pumps embedded in them, and we talked about the sodium-potassium pump when we looked at active transport. So the sodium-potassium pump is setting up a gradient of sodium and potassium and pushing them against their concentration gradients where they're going to be more crowded. So with every round of the sodium potassium pump, we're pushing 3 sodium ions outside of the cell and 2 potassium ions come into the cell. And that's going to keep the sodium levels low in the cell compared to outside. It's going to keep the potassium levels high inside compared to outside. And this is really important for how nerve cells are going to be able to send their messages to muscles to get the body to do something. Genetic disorders associated with nerve cells include Huntington's and Parkinson's disease. We'll talk about Huntington's a little bit more later. We've already looked at Alzheimer's, which is the most common form of dementia is Alzheimer's, where we get these plaques in the brain that cause decreased cognitive function. Red blood cells are also called erythrocytes, eryth for red and cyte for cell. So erythrocytes are red blood cells. They're the only cell in the human body that does not have a nucleus, so no DNA, they cannot reproduce. The way you get new red blood cells is from your bone marrow in your big bones, like your femur. So we can see the shape of this red blood cell. It's got this little divot in it where the nucleus has been kicked out during differentiation of those stem cells to be red blood cells. And in this blood smear, you can see these little light areas where the light is coming through the thin parts of the red blood cell. Instead of having a nucleus, these erythrocytes are packed full of a protein called hemoglobin. Heme means blood, and hemoglobin carries oxygen through the blood. One hemoglobin protein can bind to 4 oxygen molecules, and each red blood cell has about 250 million hemoglobin proteins in it. So one red blood cell can carry about 1 billion oxygen molecules. It's very efficient for its job. They're small, they're flat, they're disc-shaped, they're kind of squishy, so they can squeeze through these capillaries, these tiny little tubes, and go to individual tissues where they can take oxygen to those tissues. Diseases to associate with red blood cells, there is one I want you to know, and that is sickle cell disease. Instead of looking like a little disc, red blood cells from a patient with sickle cell disease look kind of like a crescent moon because they have a different protein shape to their hemoglobin. So sickle cell disease is a very devastating disease. Those little crescent moon shaped red blood cells can get caught on each other and they can clog clog capillaries, and it can be very painful for patients that have it. However, if you carry the disease, but you don't actually have it, so you only have one copy of the gene for sickle cell, it makes a person resistant to malaria. Remember, malaria was a little protist parasite. We talked about it in single-celled adaptations. They attach to red blood cells and they're carried to the liver where they can reproduce. Malaria is transmitted by mosquitoes, so when they suck the blood, they inject the protist parasite. And sickle cell disease seemed to rise in Africa, which is also where we find a lot of malaria. So chances are this sickle cell gene has been passed down through generations because it does provide an advantage if you only have one copy of it in areas that are prone to malaria where it started. White blood cells are called leukocytes, and leuk means white and cyte means cell. So these are white blood cells. They are part of the immune system. They've got these little pseudopods on them. You can see these little extensions here that are used to crawl along the blood vessels and to engulf pathogens like bacteria when the white blood cell finds one. Some white blood cells have proteins that allow them to memorize pathogens that they've come in contact before. These would be your memory T and B cells, and they're affected by HIV in particular. White blood cells also include platelets, and platelets are responsible for helping the blood clot. So when you get a cut, there's a signal sent by those damaged cells that triggers this platelets to start clotting together and making a patch over that cut, over that hole, so that you don't lose any more blood. Diseases to associate with the white blood cell would be leukemia. This is cancer of the white blood cell. So your white blood cells are mutated and they can't do their job. Sperm cells are also called gametes. Gam means marriage, and sperm cells marry eggs, if you will. They're going to combine DNA to give us a zygote, or the first cell of an embryo. Sperm cells have only one purpose, and that is for reproduction. They're going to carry the genetic information of the male parent in this nucleus here to the egg. And when they get to the egg, they're going to penetrate the egg and deliver that genetic information so that it can be combined with what's in the egg, and we have enough DNA to make a whole organism. In order to get to the egg, the sperm is going to use this long hair-like structure called the flagella. It's going to whip back and forth and help this sperm to swim in that fluid environment. This is a sperm smear of humans. You can see here a head and the little flagella on various different sperm. And diseases to associate with sperm cells are ciliopathy. Ciliopathy is when this flagella either isn't formed correctly, so it doesn't work, or it's not formed at all. And in ciliopathy, as you can imagine, these males are going to be sterile because they cannot deliver that genetic information to the egg. This is a sperm cell from the top, and this is if you were looking at a sperm cell from the side. It's got a little bit more pointy structure to it. Muscle cells are common to animals. It's the only kingdom we're going to find muscle cells in. There's 3 different types of muscle that animals use. Skeletal muscle, which contracts voluntarily. You can decide to move your muscles that are skeletal. Smooth muscle, we don't think about. It works on its own without us telling it to. This would be like the muscle that controls pushing food through your digestive tract. And then we have cardiac muscle, which is heart tissue, and it's going to just function to beat rhythmically and pump blood through the body. So muscles specialize to allow movement in some way. There are 2 proteins to associate with muscles, myosin and actin, and myo means muscle, so you can think of this as the muscle protein. And actin has these long fibers, and myosin we see here with these little heads. And what happens, we have these little divots in the actin filaments that are going to be uncovered when the muscle needs to contract. And these myosin heads can slip into those little holes and pull on them. And when we pull on them, we've got myosin heads pulling this way. We've got myosin heads pulling this way. So it's going to contract that muscle cell. And when we have many, many muscle cells all contracting at the same time, then you can contract a whole muscle. Muscles are only capable of pulling, they cannot push. So even when you do a push-up, it's these muscles on the back that are pulling your arm straight and pushing you off of the ground. So still, even though, yes, you are pushing on something, it's these muscles back here that are pulling to allow you to make that motion. There is one genetic disease I want you to associate with muscle cells, and this is Duchenne's muscular dystrophy, and we're going to talk about that a lot later. This is where the muscle cells, the protein in them, just kind of degrades over time, so the muscles don't work like they once did. Plants have specialized cells too that make up plant tissues, which can come together to give plants organs. So the 4 main organs of a plant that we're going to look at are roots, stems, leaves, and reproductive structures. Depending on the type of plant, you could have a reproductive structure called sporangia, cones, or a flower. You're not going to have all three, you're going to have one or the other. And we're going to talk about this a little bit more later, so I'm not going to get too much into this now. Roots are organs for plants, and organs are collections of tissues that come together to serve a common goal. And these roots help do several different things for the plant. They anchor the plants to the ground so that they can stay standing during strong winds. They absorb water and nutrients from the soil, and they help to store food. We talked about how glucose is made in the leaves during photosynthesis, and if the plant is making extra glucose that it doesn't need right away, it will send that glucose to the roots to be linked together and stored as starch, like we saw with potatoes. Roots can be further specialized by these structures that we see here. Only really worried about a couple of these. The root cap is pretty important. As the root is growing down through the soil, it's brushing up against rocks and dirt, and those are damaging the roots as they grow. So the root cap has all these rapidly dividing cells that are going to be copying themselves in order to repair that damage. Root hairs are little tiny hairs that grow off the side of the root to increase absorption area. So they increase the surface area for taking in water and nutrients. And we have a couple of others, parenchyma cells and sclerenchyma cells. And parenchyma cells are present frequently in the root cap because they help repair after injury. And the sclerenchyma cells make up a tissue called xylem. And we see the xylem here. It's this pink little cross in the root. There's some phloem here in the green on the outside. And those are two of the main transporting tissues that we're going to talk about. And there's also other tissues like ground tissue, but we're not really concerned about it. Stems are also organs for the plant. They're collections of tissues that come together to serve a common goal. And these stems provide support for the leaves. They elevate them up toward the sun to get as much sunlight on that leaf as possible. Stems can also transport nutrients to and from the root, and they have a couple of tissues that are going to help them do that. They have the xylem, which we can see here on the inside of these little bundles. And we have phloem or cork, and phloem you can see here in this ring. And this is a dicot stem, so this is a cross section of a typical plant stem. But we also have plants called monocots, like grasses, and they have a different structure. And we can see their xylem and phloem here in these little vascular bundles that are present all throughout the stem of the grass. Stems also have a waxy coating called a cuticle. It's going to help them retain water. And these epidermal cells are cells in this little ring on the outside of both of these types of stems. Leaves are another organ in plants, so lots of tissues that make them up. They are the primary site of photosynthesis, which is the process of taking radiant energy and converting it into chemical energy in the form of glucose. So plants are capable of using the energy in sunlight to link together carbon dioxide and water in order to make carbohydrates. And broader leaves are usually found in more shady environments because they're trying to maximize that surface area they have to get light when there might not be very much of it. So you can look at a single tree, and at the top of the tree, you're going to find leaves that are smaller in area than the leaves at the bottom, because the leaves at the top don't have any trouble accessing sunlight compared to what troubles the leaves at the bottom have. Leaves are out in the sun all day and they're getting really hot, and the sun's trying to suck the water right out of that leaf. But leaves are usually coated with this waxy substance that we call a cuticle, and that's going to protect the leaf from losing too much water. We also have these little pores, these holes on the bottom of the leaf. Sometimes they're on the top, but most of the time they're on the bottom. And these pores are called stoma or stomata for plural. And these are just holes to allow gas exchange. So carbon dioxide can go in and water vapor and oxygen can come out. These little holes are going to be surrounded by cells called guard cells that control their size. So when there's plenty of water available in the environment, the guard cells are going to swell up and they're going to push off of each other, and they're going to open up that stoma. And so gases can move freely to the inside and outside of the leaf. However, when there's not a lot of water present, those guard cells are going to lose water and they're going to start to shrink. And as they shrink or they flatten out, they're going to close off that stoma and that's going to prevent water loss from the plant in times where there isn't much water available. Of course, this is also going to prevent gases from moving between these the inside and the outside of the plant. So droughts are really going to slow down photosynthesis. Xylem is a tissue in plants, and tissues are groups of cells that come together to achieve a common goal. And these tissues come together to make up organs. So we've seen xylem as a component to our different organs in plants. It was in the stems, it was in the roots, it's also in the leaves, although I don't think I mentioned it. But xylem means wood, and xylem is going to become wood in our larger plants like trees. Xylem has one function. It's going to carry water and some of the minerals dissolved in that water up a plant, and that's all it can do. Xylem carries water and minerals up. Okay, can't go both ways. Xylem uses these cells called sclerenchyma cells, which are dead cells at maturity. It also uses these elements called tracheids. But these cells are dead. They are hollowed out and they make a tube that's going to run from the root all the way up to the top of the plant, and the water is going to move through that tube. Phloem is also a tissue that we see in plants, and tissues are groups of cells that perform a common goal and they can come together to make up organs. So we saw phloem in our roots and our stems, and it is also in our leaves, but I forgot to mention it there. So phloem is like xylem in that it can transport materials, but it's a little bit different. Xylem could only carry water and dissolve minerals up the plant. Phloem can carry water and sugars up and down. So phloem can take this glucose made in the leaves and transport it to the roots, or it can take water from the roots and transport it to the leaves. It can go both ways. Phloem also has cells that are alive at maturity. And remember, we looked at xylem, it has cells that are dead upon maturity and still do their job. So these cells in phloem are called parenchyma cells, sieve elements, and companion cells. And they're going to help this tissue move water based on the sugar content in those cells. So the sieve elements have no nucleus. They can't really control what's going on in the cell or in their environment, but the companion cells do, and they're always partnered together. So these companion cells will fill these sieve elements up with sugar, and salt and sugar suck, so it's going to fill that sieve element up with water. So here we have an example. We have a companion cell here, and it's going to funnel lots of sugars into this sieve element, and it's going to pull water into this cell. Well, if we need that water to move up, then this companion cell is going to fill this sieve element with even more sugar, and that's additional sugar is going to pull the water from this cell to this cell. And we can keep doing that all the way up a plant and transport that water to the very tippy tops of big trees. So that's all for cell differentiation. I hope you did learn a little something about how multicellular organisms get all of these different types of cells and what some of those different types of cells are capable of doing. I will see you next time.